Today on Surfing the NASH Tsunami.
But I think personalized medicine is going to change the way we practice medicine with a completely new class of drugs we call RNA interference that always has to start with genetic discoveries, but now we have the tools to silence certain genes and even modify different types of RNA that can regulate different genes in our bodies.
We need to develop drugs based on whether we think that this is a disease of select group of individuals or we think it's a population disease, because the approaches are different and the way that we conceptualize treatment is very different.
We sit here week after week discussing this. Millions and billions of dollars are being put into it. But the one thing we do know that works, irrespective of genotype, phenotype, is diet and modifying actually the one thing that people have access to themselves.
This week, close to 175 stakeholders from across the global fatty liver community have convened to hear 43 speakers address a range of NASH and NAFLD issues at the 4th Global NASH Conference. Join hepatology researchers and key opinion leaders Drs. Naim Alkhouri and Ian Rowe, liver wellness advocate Louise Campbell, pricing and forecasting guru Roger Green, and GenFit global diagnostics leader Suneil Hosmane as they discuss some of the most exciting and forward-looking concepts covered at today's NASH Congress on the Surfing the NASH Tsunami podcast.
Those with an interest in NASH, or more broadly, fatty liver disease, Surf's Up. Season 2, Episode 22, Day 2 of the 4th Global NASH Congress starts now. This episode was supposed to cover all our commentary on Day 2 of the 4th Global NASH Congress. However, we got into so much conversation that we have decided instead to split this episode into 2 parts. Today, we will cover through the point where Naim Alkhouri needs to leave the conversation to go treat patients, and we talk about some medications, we talk about genomics and transcriptomics and personalized medicine and some issues around exercise and diet as well. It's really a pretty broad sweep for 38 minutes. Saturday morning, Conversation 1 will be the second half of this episode, where we talk a lot more about drugs and about exercise and diet and other influences that affect how we will be treating patients going forward. I will now return you to the original episode. Have fun, have a good listen, and remember to come back Saturday morning for episode 23, where we will finish covering the conference. Thanks. Literally, Day 2 of the 4th Global NASH Congress wrapped up maybe 10 minutes ago. Much of yesterday's program focused on innovative diagnostics. Much of today's focused on drugs in development. There were other topics of interest that Ian and Louise at least will cover, but some of the presentations today were simply recaps of prior research, while others were really novel and different. We'll get to all that in a minute or two. Manal's not here today, Yorna's not here today, but we're fortunate to have with us Naim Alkhouri. Hey Naim, how are you this morning?
Good, how are you?
Excellent, thank you. Excellent. And Ian Rowe. Good morning, Ian.
Hi, Roger.
And of course, Suneil and Louise are back. Again, schedules are tight between attending the conference and the normal workday, so we're going to try to dive right in. Let's move on to today's conference. I want to use the same icebreaker we used yesterday. One thing you heard in this Congress that got your attention or was a little surprising, and why that is. Bonus points if it's not on a presentation that you personally are discussing today. Brave one, go first.
Well, I guess I'll go first, Roger. So I think, we listened to several presentations, different pharma companies, very attractive, therapeutic targets, very encouraging preliminary data in terms of efficacy. We have a lot on safety. But the thing that, I'm noticing more and more that, in each trial, we have all these non-invasive biomarkers, whether they're serologic or imaging. And, I think we will have very robust data in the near future. And, much of it will be in comparison to biopsy changes over time that will hopefully, if these pharma companies collaborate together, we can maybe move away completely from liver biopsy in terms of trial design and endpoints. My surprise is, why aren't we doing this now? I think, this is a message we need to drive, that we need several pharma companies to collaborate on this, because the end result will help everyone in the field. And, we see it in almost every presentation, whether it's MRI-based or serology-based. I think the time is now, before we have actually FDA-approved drugs in a couple of years, years to figure out that pathway in terms of identifying high-risk, juicy NASH patients, as Stephen always says, and then to figure out a way to monitor them in terms of response to treatment, determine futility, the need to switch to new agents, and how we can prognosticate them. So this is probably my take-home message listening to all these presentations.
That's a great place to start, Naim. And because we're on audio and therefore our audience can't see us, I will tell them that heads were nodding as you were saying that, bobbing up and down, somewhat enthusiastically, in one case quite enthusiastically.
Mine was from a presentation by Shira Zalbayasagi, who did the nutritional epidemiology to practical nutritional treatment in NAFLD. And the thing that surprised me most, and it probably follows on from Suneil last night being controversial, was she reviewed the placebo response in randomized controlled trials and pharmacotherapies and noted that 25% of patients given a placebo improved their NAS by more than 2 points, and that 21% of patients given a placebo improved fibrosis for more than 1 point. So just being slightly controversial at the start, following on from Suneil saying maybe we're expecting too much from the devices and non-invasive that we're doing, and maybe they're measuring the wrong thing. Maybe we should start any placebo response at 21 and above for a trial and accept that as a minimal standard that they've got to tick, which means we prove efficacy on top of that. So we set a benchmark.
Louise, just to say something about this, if you take the baseline biopsies in any trial and then have the same pathologist just read them without any intervention, they'll give you 20% improvement in the NAFLD activity score. The same pathologist reading the same slides. This is the reality with biopsy. It's a very unfortunate situation we're in, that we have this field, billions of dollars, all these pharma companies, and we're relying on pathologists reading slides. I mean, they're great colleagues. We love our pathologists, but it's not fair to anyone what we are dealing with.
So that's an interesting question, right? So that raises the degree to which what Dr. Zalbayasagi was talking about is an artifact of how slides get read versus a reality of placebo. And I'm not even sure how we answer that until we get past slides, but it's an interesting point.
Well, I'll go next, because what I'm going to say sort of connects both of those thoughts. In the first talk this morning, Sven Frank talked about the concept of advanced chronic liver disease, which is fibrosis stages 3 and 4 combined, as being prognostically significant liver disease. And the Bovino consensus, which looks at advanced liver disease and portal hypertension, would allow you to define that using transient elastography. So that begins to push you towards a trial entry point that's defined by elastography and is defined by prognosis, so that you can begin to think about how your treatments will affect an impact on patients in the hopefully not too distant future. So I thought that was quite an interesting idea, and it's the first time I've heard it articulated in terms of trial entry.
It's a fascinating question, and the question I posed that either I posed so badly that it didn't get answered, or the folks from Global Engage never saw it, was a question to Xiaolu about why they're enriching F3 patients in their phase 3 trial, and whether that in fact had anything to do with those issues. I mean, they're doing it because they're trying to get better response, but whether in fact they thought in the process they might do a better job of actually honing in on patients who need the drug most badly. It's an interesting question, whether we rethink a lot of that. We'll come back to it later. Suneil?
That's an interesting thing, actually, that I have some thoughts on. Maybe we get to that never part of the podcast, but Suzanne Norris had a wonderful talk. In fact, I'm sad to say I missed the last 3-4 minutes because it went over a little bit and I had another meeting I had to get to. But I love when people take concepts that we know, but they quantify it, because it just kind of elevates kind of what it is. And so one of the things that stood out to me was 7% of the EU population exercises, does active, regular exercise. I thought that was incredible. We all know it's low, but my goodness, 7%,
wow.
That's
wow. Yeah.
And then she started getting into the details of different, I don't want to say interventional trials, because it's not a therapeutic or anything, but it's exercise or exercise plus resistance training, rigorous, looking at, a certain period of time, 12 weeks, and sometimes even biopsy at the terminal point and the baseline, and really showing that, it works. 58% saw, I don't like giving out just numbers without the context, but 58% saw one-stage improvement in fibrosis with very marginal change in BMI BMI, and that led to another point, which we all know, but we don't talk about as much as BMI is a very crude metric, and so a lot can happen in the body, and the BMI stays the same, right? And we know that's true. I mean, I know that any of you follow combat sports, right? You know the severe weight cut that occurs, right? So these people are having massive BMI changes over a short window of time, but it's water, and so what's happening on the inside matters, and we shouldn't necessarily stigmatize like just pure weight, but becoming a healthier version of ourselves, which then links to other things that people have spoken about, which is crudely one could think maybe the best marker, the easiest biomarker is BMI or weight loss, right? Because it goes up, goes down, and we can easily measure it. You can do it at home. But if you combine it with this concept, you could be changing dramatically. A lot of things could be improving. Liver fat could be improving. A lot of inflammatory processes in the liver could be improving. But you're not going to see it on the scale, right? So what sort of biomarkers or things could we use to help identify whether patients are getting healthy, because it's needed. I didn't realize that the need in the non-therapeutic area, but it's just as significant as the therapeutic area.
That's interesting. I'm going to go into a paper that I think Ian's going to talk about from a different angle. In the States, I think the VA does not normally get high marks for the quality of the health system, but I was really impressed by Michael Fuchs's paper today and the amount of thought that had gone into putting together a complete NAFLD plan for the VA. They've thought everything, pretty much, and they're 5 years out. And I don't recall whether this was in the Q&A after the session or in an offline conversation I had with Michael after that. But one of the things he talked about was having a challenge getting, I think yesterday in a Q&A, about getting fiber scans out to rural populations. So I asked him how he tended to deal with that. But a remarkably well thought out answer, and the most impressive thing I've ever seen come from the VA. I thought it was just fantastic and really well put together.
I think the VA system in the U.S. has been helping us answer many important questions in hepatology, whether it's related to hepatitis C, elimination, HCC, surveillance, and now NAFLD. So kudos to them, but I think, they generate a lot of very important data for the whole field.
In hepatology, they've been fantastic. And as he noted, they were the first health system to eliminate Hep C, because obviously the government could buy DAAs cheap, fast. But yet, no, on hepatology, they've been great. Okay, so with that, let's go into today's activity, which is talking about papers from the conferences. And we'll do what we did yesterday. Take the 2 papers or 3 papers that you were going to cover and tie together a theme or not, and go through what you think were the high points. Take 6, 7, 8 minutes to do that, so we have time to chat before we go on to the next one. Naim, I'm going to suggest that you go first, only because you're the one who might have to exit early. So why don't you kick us off?
Appreciate it. I selected 2 presentations during the meeting that I thought, they're important. First one is by Stefan Romeo, talking about the genetics and personalized medicine in NAFLD. And, I have a special interest in this topic, but I think this is going to change, the way we practice medicine with the completely new class of drugs, which is, what we call RNA interference, that always has to start with genetic discoveries, but now we have the tools to modify gene expression and silence certain genes, and even modify, different types of RNA that can regulate different genes in our bodies. So, he started by discussing the discovery of PNPLA3, the most validated polymorphism that contributes to the development of NAFLD, but more importantly, its progression to advanced fibrosis, hepatocellular carcinoma, and end-stage liver disease. This gene obviously plays a role in other liver diseases, including alcoholic liver disease. He also provided some data on all the other variants, including TM6SF2 and MBOT7, and then some of the protective variants like HSD17, beta 13. And then he provided a deep dive into the PNPLA3 role, including, its effects on the lipid droplets in the hepatocyte, but also on the hepatic stellate cells and, its effect on liver fibrosis. He provided nice data on the prevalence of the M allele and its importance in terms of predicting what patients with NASH will end up with cirrhosis and HCC. So I think, this was kind of like an overview. I think, what's exciting for me is that now we moved beyond the discovery of PNPLA3 initially in the Dallas Heart Study and the validation in several cohorts to establishing its role in disease progression. And now the last step is how can we modify PNPLA3 expression to basically change the natural history of the disease. And we've seen some preliminary data on that. I think this is an area that but as I said, it's going to change how we think about different chronic liver diseases. And there are several genes that we can modify in the context of NASH. We have drugs that are FDA approved today in the United States to target other diseases that start in the liver, such as hereditary transferrin amyloidosis. This is a disease that I've seen several cases. The mutant protein is produced in the liver, but actually the disease manifests in the neurologic system in the form of ascending neuropathy in terms of heart failure, and you can have also this autonomic dysfunction. And we can silence basically the gene and decrease the expression of the gene and protein production, and in the way help mitigate this very progressive and life-threatening disease. We have drugs that we can use for acute intermittent porphyria now, and the field is expanding. So there are trials looking at alpha-1 antitrypsin gene silencing also. We know that the mutant protein is produced in the liver, it functions in the lungs, so if you don't have it, you end up with emphysema, but also liver cirrhosis. We're looking at even hemochromatosis and potentially silencing some genes that can inhibit hepcidin. And of course, the holy grail is looking at liver fibrosis and see what genes we can silence with RNA interference to improve liver fibrosis or decrease its progression. I have been involved in some studies looking at heat-shock protein 47 and using a short interfering RNA to downregulate its expression. So what he provided was kind of an overview of the evolution of genetics in fatty liver disease, provided several targets that we can potentially hone in and develop new antisense oligonucleotides or short interfering RNAs to target these genes. And I think this is going to be part of the armamentarium that we have to treat NAFLD. So that's the first presentation. The second one, I think more practical, the galectin presentation by their CMO Holrood. So he presented initially a lot of background data on the role of galectin-3 and liver fibrosis, potentially its role also in the tumor microenvironment, and they do have a program targeting HCC. He presented some animal data, how belapectin decreased macrophages in the liver, collagen expression, deposition, decreased portal pressure, and then he went on to share the details of the published paper that we were part of, where they randomized patients. It was 162 patients randomized to the high dose belapectin 8 milligrams per kilogram, low dose 2 milligrams per kilo, and then placebo. I mean, the results have been published. This is not new data. I think they did not meet the primary endpoint in terms of hepatic vein pressure gradient reduction. But what they found in that trial, that you use the lower dose, actually the 2 milligram per kilogram, in patients without varices at baseline, that these patients were less likely to develop varices at the end of the trial. And if I remember the numbers correctly, in patients in the placebo arm that didn't have varices at baseline, about 18% developed varices. If you look at patients on the 2 milligram dose, it was at the end of the study 0% or none of the patients developed varices. So that was interesting finding, and that's why this adaptive phase 2b3 trial that we are conducting now selected patients with evidence of NASH cirrhosis, portal hypertension, but no varices at baseline, and the end result is really the development of varices. So this is kind of like an endoscopic endpoint for a NASH trial, which is, to me, is innovative and exciting. Obviously, they selected kind of like a post hoc analysis. They found a subgroup of patients where the drug worked. They selected one dose where they saw benefit. So, you have to take all of these data with a grain of salt, but I think, this is the biggest unmet need. I know Ian loves to drive the point that we need to focus on patients with F3, F4. This is the highest unmet need, but within that really advanced population, patients with NASH cirrhosis and evidence of portal hypertension is even a highest unmet need. Of course, it's a higher bar, and showing benefit in this population is definitely more challenging, but I think if we can start, targeting these advanced patients, that would be great for the field. I think, NASH cirrhosis trials, this is an area that's witnessing a revolution in a way, and a lot of interest, especially after the FDA webinar and the whole pathway to drug approval, where you can just show outcomes with NASH cirrhosis, and the fact that for F2, F3, you can just do a histology study. Having safety data in patients with cirrhosis is very important for any NASH drug development program. So I'm very interested in this area, and I thought, Paul did a good job taking us through the whole development process and sharing the mechanism of action, animal data, preliminary data, and then future plans.
The idea of highly personalized genetic-driven treatments in a common disease is an interesting one. And I know that I go on about primary prevention of coronary heart disease all the time, but it's interesting that mutations and relatively common traits in coronary heart disease that haven't been drugged because there are, not particularly effective at the individual level, but highly effective at the population level preventative strategies. And I say it often that we need to decide and develop drugs based on whether we think that this is a disease of, a select group of individuals, or we think it's a population disease, because the approaches are different and the way that we conceptualize treatment is very different. Whether there is a place in therapy for a drug that's going to be effective for a small number of people remains an open question.
Ian, when I first came to liver disease, one of the things that struck me, this is a sign of how old I am actually, is that in 1969, Nixon, then president of the U.S., declared the war on cancer. And at that point in time, the big concept was the drug that cures cancer. And what we learned over time was not only was there not one drug that cured cancer, but there was not one drug that cured breast cancer. There was not one drug that cured colorectal cancer. And it's by diving further into this and understanding the complexity that we were really able to get to solutions that were solutions. There are reasons that NASH has struck me the same way, lean NASH being one of them. The idea that you got 6 or 7 different modes of action, each of which work 30 to 40% of the time better than placebo. The idea that you get so much spontaneous regeneration. I think there's going to be a creative tension or scientific tension or some form of tension for years between the two sides of that argument until either someone comes up with the drug that cures NASH, or we realize there isn't going to be one because there are 6 drugs, each of which do really well, but not in more than 10% of the population need. I think it's early to bet, frankly.
It's such a complex question. I had a friend, I asked this question in a related field, like GLP-1s, right? We all know GLP-1s do wonderful things, tremendous data. Whenever you see the data, like just for like the obesity side, you see like 15% weight loss on average, right? No one's experiencing 15% weight loss. You have a population that's experiencing higher, and then you have a small group that's experiencing 0 or very small, and you kind of average out, and it's 15%. And from clinicians, what I've heard is like it's about, maybe it's a little bit high, but 25% of the population doesn't respond to the GLP-1. And so you think, would it be valuable to have like a diagnostic? You could kind of understand either the non-responders or the responders, and you can have a kind of a slightly better tool that's going to get the 25 or the 75%. And a friend of mine, I've asked that question to both Novo. I don't think they realized what I was asking, but I asked them that at one of their events. And as all of their strategies, biomarkers, companion diagnostics is not on the roadmap. They don't, this is not something that they are looking into. It's not something that they care about at the moment. So that's just an interesting, piece of information, I think. And then my friend, who's been in the commercial world for many, many years, she's an exceptional marketer, commercial person. I said, wouldn't it be cool if you could do that? And she's like, the flip side of it is, are you now introducing like doubt to the physician that this thing can't work or it may not work? And does that create now additional complexity that you're not thinking about that will surface when the product goes to market? So I think it's a really, really awesome question. I think it's an area where we have to have some footing in NASH because we know there's a genetic component, but we know there's a huge environmental component. And maybe there are subgroups where that genetic component, and that's it manifests as NASH, but it's a very different disease. It has a unique kind of, therapeutic strategy, and maybe the bulk is kind of a mass market thing, and you have, like the point was made, you have a different strategy for that too. So I don't know the answer, but it's an interesting question.
My only other thought was, we sit here week after week discussing this. Millions and billions of dollars are being put into it. But the one thing we do know that works, irrespective of genotype, phenotype, is diet, and modifying actually the one thing that people have access to themselves. And in the dietetics presentation that I saw that I referenced earlier, there were a lot of people that just the quality of diet changes significant steatosis without weight loss. So picking the right phenotypes that are going to respond to the right TARS of diet. But actually, all humans seem to just want to do what we want, take a pill, but it comes with side effects, rather than go for a simple resolution, aka Suneil's comment that only 7% of Europeans exercise and 47% I think have never exercised. So
we can start at the society level and talk about the food industry history all the way to treating patients with NASH cirrhosis and portal hypertension. But I think we need every piece. I don't think we need to say, oh, well, it's just a diet problem, because we don't do that for type 2 diabetes. I don't think we also need to just focus on really sick people that will die from liver-related disease, because then, we would wait on treating dyslipidemia until you have evidence of coronary artery disease. I think many people, especially in the US, we have the concept of wellness also. I don't want to have an illness. I don't want to have NAFLD even, right? Many of my patients, I tell them, oh, I have good news for you. Your FibroScan shows, you have fatty liver disease, but you have no fibrosis. You're good. And they look at me like, this is terrible news. I don't want to have any fatty liver disease. Same thing with dyslipidemia, same thing with insulin resistance. These are all, they're not diseases, right? They are risk factors for a disease that could be potentially life-threatening. Of course, liver disease, because the liver is so resilient, hasn't been a major killer, even in patients with type 2 diabetes. But the concept of wellness, I think sometimes we lose it because we're struggling with no medications and we want to treat the sickest patients first. But honestly, this is what we want. We don't want to start with illness. We want to keep people well.
So, Naim, 2 or 3 comments, of which I'm going to reserve one for myself, and then let's move on to the next presenter. When you mention dyslipidemia, I think to some degree that is the model of what we're talking about, right? Which is if you're mildly dyslipidemic, if your cholesterol's elevated and it's the only risk factor you got, then you go to diet and exercise. Beyond that, if it presents a problem, if there are other risk factors, you tend to go to a statin. But unless somebody's had a couple of coronary events, you'd have to have pretty high cholesterol or even familial before anyone takes you to something really expensive like a PCSK9. I could envision something similar here. That I think you're right. Portal hypertension, decompensated cirrhosis, people are rapidly approaching death with high confidence that if you don't do nothing, you're going to get to a bad outcome. We'll spend more money. We'll look for more intricate solutions. Earlier on, it would be great if we get the society to exercise. We were talking yesterday about fructose metabolism and what would happen if you could simply reduce fructose consumption, right? But that's probably not going to happen. So just multiple levels of attack. Other thoughts?
This is unrelated, but I was watching a documentary on Netflix about avocados and just realized that to grow one avocado, you need 18 gallons of water. So everything is complicated, Roger. Once food supply and there's no simple answers. I think, so we need to accept our role as physicians, and then diet and exercise should be part of any strategy anywhere. There is no question. This is very important. This is the best thing you can do, but we need to be prepared for failure of diet and exercise and what to do next. But I'm a big, proponent of, physical activity, eating healthy, working at a society level in the schools, policy, thinking globally also, because the, global economy is what drives prices and policy. So it's complicated, but as a physician, I don't like to get overwhelmed with the global picture. I focus on what I do best, which is identifying high-risk patients and treating them and preventing bad outcomes.
Well, thank you for the avocado fact. I'm sure that'll get used at some point in my life in the next 2 weeks.
I'm actually going to jump in here purely because I did Anne Daly's genomic and transcriptomic analysis of NAFLD, which obviously follows on from Naim's presentation, but I won't do it as much justice. So basically, she summarised genetic susceptibility to NAFLD, transcription analysis of NAFLD and tissue from biological change to see if we could look at disease progression and can we predict susceptibility to NAFLD. So they did studies of genome-wide association studies, or GWAS. They included over 1,000 cases and suitably matched people. And they took from the FLIT and EPOS studies in Europe. So they had quite a number of patients. So they obviously looked at the modifier genes, PNPLA3, they did discover was quite high. They also went for all of the other studies that had shown that GCKR, TM65F2, chromosome 19, and then BOAT7 were identified in their studies as well, but also alcoholic liver disease obviously has BOAT7. So they went on to look at 1,483 NAFLD cases. They had a central data population of 17,781, which were taken from the UK, Germany and Italy, so they had a good population. And they had 7.4 million SNPs and genotypes. So they found 4 significant signatures with p-values of 10 to the -8. PNPLA3 the TM65F2, the HSD17B13, and GCKR. But they had several near misses, but as they got further down, they found that some of these were protective, and the near miss of relevance was the MARC1 and MTARC1 SNPs. So putting that into practice, the subgroup NASH were very much PNPLA3 and leptin receptors and for signals, but only the F3, F4 had the PNPLA3 and TM65FS and GCKR. They didn't have the others. So when they went on to the transcriptomics, the biopsy samples, they had 206 samples. 40% of these were female, average age of 54 and a BMI of 31. But over 55%, just over, had type 2 diabetes. And all fibroses were represented from 0 all the way through to cirrhosis. But the key findings, they were able to find a 25 gene pool that was relevant to these specific patients. And most of those, 24 of those were highly positive and progressive, but only one decreased, and that was the HSD17B14. And this was replicated when they did replication studies. They then went on to the microRNA, which was relevant from yesterday's sessions. And the main microRNA of choice was the 193A5A, which was replicated in NASH with fibrosis 1 and increased in fibrosis 2. So the general comments at the end with all the risk factors are fairly similar. Gene expression changes, specifically at least through progression, there were quite a number of gene changes. And the microRNA was interesting for understanding, but it also could assist in the diagnosis and staging of the disease as we progress. So I thought that was quite an interesting topic about being able to, again, genetically identify people who are likely to progress, which may be more relevant when we start to locate these people. So that was because it connected to Naim's. But my second presentation was Heather Bryson, who was Executive Director of Accelerated Enrollment Solutions, and she was doing and reporting back on a study of awareness of non-alcoholic steatohepatitis and treatment guidelines and its progress being made. And I think I remembered seeing this first survey. They did a survey of physicians from all backgrounds in 2019, but it was predominantly US only. So they repeated that survey in 2021 globally, and they're still collating the data. So these were early results, basically looking to identify what has changed in practice, what the unmet needs were, what the barriers are, and whether or not there's an increased awareness of what's going on. And it was interesting. They sent out polls to 18,321 physicians, mainly primary care physicians, hepatology, gastro, endocrinology, and some others, but only 96 had responded. So this survey is based on relatively few numbers, but they are still collecting the data. And understanding of NASH and NAFLD really haven't overtly progressed. The differences, and in fact, it was slightly down, which was a concern in gastro, but only markedly. But they were still the set of physicians that had more knowledge, which we would expect that to be the case. Increase slightly in primary care physicians. The diagnostic measures that people were doing, which is of a serious concern, there were a lot fewer blood tests, although people were using more imaging with blood tests, but biopsy was down, more or less 2/3 in every one of the categories, people not referring to liver biopsy. And the reference tools that were being used, FibroScan has increased significantly. 86% of endocrinologists surveyed were now using it. FibroScan with 90% of gastroenterologists, and primary care physicians have gone from 39% to 60%. So obviously there is increasing access to those patients. But not sending, one of the primary risks for not sending most patients for liver biopsy, and they'd more or less reduced by 60% in all of the categories, as I say, was that they didn't see that the risks outweighed any benefit, and it didn't make any difference to their treatment plans. When we looked, they had a slide that looked at the COVID impact, and it's concerning. We look at clinical trial recruitment, 60% fewer patients are receiving blood tests now to monitor liver function tests, 88% fewer patients receiving liver biopsy. And obviously liver biopsy is the gold standard for NASH. To be able to recruit into studies, we need these patients coming through, and this is our pipeline. 72% fewer patients receiving non-invasive tests, liver imaging such as FibroScan or MRI, despite them being going up. 83% of primary care physicians refer fewer patients with possible diagnosis for NAFLD and NASH, and 75% of specialists receive fewer referrals. So if we're not seeing these referrals, if we're not getting the knowledge increase, there really is a problem for future recruitment of clinical trials, which was the ultimate outcome of the findings that we cannot recruit to clinical trials. And we've seen a 35% of sites that can't recruit a single patient in NAFLD and NASH trials. So there's a serious problem, and it's only just starting. We think we've had a problem over the recent last year with COVID. Well, COVID's affect the pipeline for the future. So that was the summation of, based on a few patients, referring physicians coming in, but it's a concern.
They got how many responses from sending on the second study? How many responses and how many did they send out?
They sent out 18,000, and they only had 96 when they did the survey, but a lot. And
not 9,600, 96, less than 100.
96. So it was very, very small, but it was the US physicians only they were comparing, because, of course, the first survey was US only. The second survey was global, so they were only able to take those 96 that were US only. But it's still a concern.
It might be a bigger concern because I don't know why the people who didn't reply.
They are replying, but this was done quite early because this is a 2021 study. We're now only into April, so it was really looking at the effect of the pandemic. So I would hope anybody listening responds.
What I hear you saying is stay tuned or watch this space. Other thoughts, comments on either of the two papers that Louise was talking about?
It's nice to see more NITs being used. When you said that we're doing less biopsies, I think that's music to my ears. It's okay. We can live without a liver biopsy. We are struggling really with clinical trials. I'll give you an example. Just yesterday, I got this report on a patient for a clinical trial. This patient has F3, on the biopsy. They have a NAFLD activity score of 3, 1 for steatosis, 1 for ballooning, 1 for inflammation. Now, this patient with F3, with ballooning, inflammation is not going to qualify for a clinical trial because the NAFLD activity score is less than 4. This same patient has a liver stiffness of 13 plus minus and elevated ALT and family history of NASH cirrhosis. I mean, it doesn't get higher risk than this. And now I cannot include in a clinical trial. And my worry is that, fine, clinical trials are the tip of the iceberg. Not everyone is open to the idea of a clinical trial. There is a placebo. But once we have effective therapies, if we keep doing biopsy at baseline to qualify patients, if insurance companies just go by the trial design and say you need to meet all these criteria, if we keep driving that point that, activity initially predict fibrosis response and you need to have, 4 for the NAFLD activity score, it's going to be a struggle. And I'm not doubting the science behind, the importance of NASH as a driver for fibrosis. What I'm doubting is, the pathologist reading the biopsy and maybe underscoring steatosis. And we all know how semi-quantitative it is. And if they decide, maybe it's 30% steatosis, they give it a grade 1. Next morning, the same pathologist can look and say, maybe it's 35%. I'll give it grade 2. I mean, this is the reality. Or you miss, a ballooned hepatocyte here and there, which is very common. So I think, we need to just move on to, finding a way to identify high-risk patients. I think the fact that we're doing more NITs, FibroScans, I mean, it's all good, but we have a long way to go. The issue with these surveys that usually the responders are the people that care about the disease state they're being asked about, so they tend to be more, proactive. The non-responders are the people that they still see the same patient population, but they just don't care to take the time and respond. So, these results are typically biased toward more aggressive management.
Naim, I think you hit that nail on the head. I've got nothing to add to that, personally.
I think the one thing that surprised me in the information that she gave was that clinical trials take colossal investment to set up, yet 35% of all trial sites cannot recruit one patient. Does that lend to the due diligence of these trial organisers, or is it sites pitching for something they don't and can't deliver to?
Naim, let me do an answer that's a little bit broader than you, which is that NASH is a tough recruit. And I hear that everywhere. Okay. And if you look at the AES business model, is they look at sites that do lots of different things. So it's not surprising to me at all that 35% of if AES was surveying the sites that they deal with, it's not even remotely surprising to me that 35% of them can't do NASH. In fact, I think that might be, that feels to me almost like an optimistic number, certainly not a pessimistic number. If that was the sample, I don't know who the sample was, but if that's it, that would not surprise me.
Yeah, I mean, we have several steps, I guess, because that's what I do day in, day out is clinical trials. So, Louise, it's so easy to exclude someone from a trial. If you change your diabetes management, go up on the dose, add a GLP-1, automatically you have to stay stable on these medicines for 3 to 6 months. If your A1C is above 9.5, if your BMI is above, it depends on what trial, but sometimes as low as 40, which is difficult for us in the US, we cannot include you in a trial. Then you move on, and then you have, your MRI PDFF, and, you can be at 9.9 and be excluded from a trial. And then after we do all this, we do the biopsy. We think we found, patients that are suitable, and you still, if you know what you're doing, you still screen fail on biopsy in 35% of the cases. So the screen failure rates in NASH trials, at a very good site, if you're at 60%, you're considered like standard for industry. And that's why you have sites that remain at 100%. I think they all screen. They cannot find suitable patients. And, also having experienced coordinators, people that know how to ask the initial questions quickly and just exclude patients, because it's a waste of everyone's time if you bring someone to do a screening visit to find out that they just started metformin a week ago. So these are, quick things that you have to do. Once you do it day to day, you get used to it. But it is frustrating. I think what's saving the field is, the large number of patients. If this was, something that affected, 1% of the population, we would be struggling big time.
I can just make a small comment because I was on the other side, and I've seen the site selection process and things like that. I would say it's both. So, the two reasons that you mentioned, it's a little bit of both. I would say most sites and most clinicians are doing their best, and you can see that because you see all the logs, right? So you can see the screen failure, the screening rate, and you can see how many screen failures versus randomized. And I don't know, but I can infer when you start to go through a bunch of screen failures. You talk about like willpower and someone breaking your back. You may give up, right, at some point if you have a series of tough times identifying a patient. You may slow down, and I don't blame anybody for that. That's like fatigue. That's going to happen. A small part of that blame also goes to the pharma company, because they're under immense pressure. They want to enroll, right? They want to find sites. Sometimes people don't want to work with you, right? For various reasons. They're filled to capacity. They're doing other things, and you sometimes you cut corners, and you think that you have unrealistic expectations of what someone's trying to do. And when you get there, generally people are very optimistic, so they're going to give you a little bit a higher sense of what they can accomplish versus like, oh yeah, I can just get one thing done. So, and some of those sites end up not really understanding what they're getting themselves into. And a consent for a trial phase 3 is like 14 pages. As you go through it, you may talk yourself out of even being interested in doing this thing, right? Which is going to manifest in how you communicate with the patient. So some of it is difficult. There's fatigue. I've heard that comment about the biopsy a million times. I know it's true, and then I also know that sometimes people just don't know what they're getting themselves into. That they have a desire to do it, but once you see all the nuances to clinical trials, it's not for you, and that's fine too. But it results in enrollment issues.
Okay. We're at about 12:30, which means we lose Naim in just a minute, and everybody else, I think, shortly after that. And we still have 2 or 3 of us to go. So, Naim, any closing thoughts before you go treat patients and do the day job?
No, I feel like I spoke too much. I'm going to shut up here.
No, it was great. It's been great having you. Step out when you have to, okay?
I'm curious to hear, what others have to say. But if I have to step out, it's just because I have patients, so apologies. Okay. And then I will listen to the whole podcast when it's posted. Thank you guys so much.
This ends the first half of our coverage of Day 2 of the 4th Global NASH Congress. We will be returning with the second half as episode 23 on Saturday morning. Till then, let me thank all the surfers who participated: Louise, Suneil, Naim, Ian. Let me thank Mike Wilson, who once again is making magic happen before our eyes by putting high quality sound in front of us, and Eric and Polly, who've had to adjust for this change, and all of you, our listeners, who motivate us to do the best job we can and put the best product out every time that we come to you folks. Finally, I want to thank everybody from the 4th Global NASH Congress, most particularly Nick Noakes, who's been so easy to work with all the way through this. Thank you. We'll see you Saturday morning, and until then, stay safe and surf on. See you on the podcast. Bye-bye now.
Have any questions for the surfers? You can send them to questions@surfingnash.com, and we will answer on Monday's wrap-up podcast for the 4th Global NASH Congress, or on the website. Join us again Saturday for the second half of our coverage of Day 2 of the 4th Global NASH Congress, right here on Surfing the NASH Tsunami.